Good morning, everybody. So our final speaker for the morning session, I'd like to invite Clint Valentine to the podium. Clint's Senior Director of Computational Biology, and he's one of those modern scientists who's steeped in biology and also in data analysis and computational methods, which are sort of de rigueur for pushing this science forward, I think. And he spent some time at MIT where he helped pioneer duplex sequencing. So it's with great pleasure to have Clint come up to the podium.
Yeah, it's really great to be here, and these talks are awesome. I'm loving everything so far. This will be a unique complement to everything we've already heard about today. I'm going to take what Jesse and Inigo talked about, where they introduced the technology and then showed some applications, and draw a line between that and what Jill has just presented, in which you get to a trinucleotide spectra. So in the middle there somewhere is bioinformatics data analysis. Now we're moving from tox assays that are a little bit simpler in principle to something that actually is quite complex. And I put this up here not to scare you, but hopefully by the end of the talk you'll understand these little squares and these little squiggles. These are all just a part of basic concepts that aren't too hard to understand. So like Bob, I'm going to talk a little bit about history of myself and the journey that I took to get here. How does this work? Oh yeah, here we are. So I started my career actually not in biology or in data science or anything at all. I started as a machinist and an engineer, and I was really fascinated with how systems worked, how machines were built. I drifted into bioinformatics after that. I wanted to go to school and became a genetic toxicology researcher at MIT. And I worked at a lab that did a ton of Big Blue, or actually we didn't do the Big Blue, we did TGR, but we did the GPT Delta. A lot of comet chip, a lot of other tox assays. And right around while I was there was when duplex sequencing was invented. And our lab was kind of like a sister or brother lab to the lab Jesse was at. So we brought the technology over and we coupled it together with genetic tox. Later on, Jesse had decided to start a company around this idea, and I just had to be a part of the experience, so I joined him. And now I lead the data sciences group, so that's about half of the R&D at TwinStrand. Oh, and that's Jesse there, we've climbed. These are all the things that I like to do personally. And why I'm showing this slide, actually there was a point, is that when I began my journey, I really enjoyed the automation of things, the making things easier and simpler on humans. I wanted to spend most of my time outdoors and do all that. So as a common thread here, a lot of what I've done is just to make all of this easier for you, to make it more accessible. So you can go outside and play. One of the first things that we did when working with duplex sequencing is we applied it to a transgenic rodent model, the GPT Delta mouse. And instead of going through the laborious path of package and phage and whatnot, we just took the duplex sequencing method and applied it to that transgene. And we were able to discover aflatoxin signatures and spectra straight from that bit of DNA. And that was really, really interesting. Not only did we get that detection event and did it look like aflatoxin spectra, but it was also detected, I think it was 10 weeks after exposure from a juvenile mice, so before neoplastic transformation. So this kind of revealed the potential for short-circuiting some of the laborious assays that we had in the past, and we're dealing directly with mutation data, so that was really interesting. It's funny, Jill showed some spectra for aflatoxin. I believe when we did the study, well, this is actually like signature 24, but the empirical one that we got from the study was like CGC was really hot, and we were like, why is that? It's a little bit different than the two you found. Then I moved to TwinStrand and began working on this in a commercial sense to make it better, to make it easier. We've done a couple publications there that resulted in one that I think is outside, that was in collaboration with many people, including Bob. Anyway, this is where I am still. So what am I going to focus on is taking duplex sequencing, applying it to genetic toxicology, and then talking about the bioinformatics and the data analysis. But I did want to highlight that genetic tox is just one part of this big span of applications that we could really work with in error-corrected NGS. So what does it mean to measure DNA mutations directly with duplex sequencing? What are we doing and what can we do with it, and how is it different than what we've done in the past? So this slide has been shown before, but we are taking a source molecule, so that's truth, that's something from an organism, and we're using technology to read it and to create a near perfect reconstruction of that source molecule. And theoretical, very, very low error rates. Existing assays are kind of on the left, the ones that I've compared it to. There's a 2-year rodent assay there in red with the mouse. There's in orange, there's the transgenic rodent assay. And then on the right is direct DNA sequencing through duplex sequencing. On the right, a little more simpler. It's also a little bit more quicker, and the data readout that you get is much more information dense. So with like a plaque assay, for example, you might be able to tell if you have mutation induction or not, but you don't know unless you go to sequence after what those mutations are and what could they mean. So you're stopped there. But if you move over to true sequencing, then you can get a lot more information from that data. So these are all the things that you can get from directly quantifying a mutation instead of measuring a proxy for a mutation. So we can look at any DNA-based species, any tissue in those species. We can look anywhere in the genome. We can characterize these mutations. So we think about base substitutions simply, but there are so many more other kinds of mutations. There's insertions, deletions, complex variants, structural variants. We can detect all those. And then we can correlate them with genomics features, like transcription status, chromatin structure, and anything else you might be interested in. And then finally, once you have all that omics data, you might be able to predict the functional disruption of those mutations in the cell. What could it mean, given all that biology above it? So getting into the bioinformatics, how do we make this happen? So you sequenced your sample, and immediately you're going to get a bunch of files. They're going to be really, really big, many, many gigabytes, and they're going to kind of look like that on the left. And they're simple, but they are large. So they have sequences that are the DNA, read base by base, along with quality scores. The quality scores are the calls that the sequencer made that predict how correct it thought it was when it made that call. And that's important because sometimes the machine isn't so sure, and we want the machine to tell us that information. That data will all go into a duplex sequencing bioinformatics workflow. And this is a schematic of a kind of a generalized one. And if you're familiar with DNA sequencing workflows in bioinformatics, it's almost identical, except there are a couple extra tasks that you need to be aware of, specifically around extracting the UMIs, perhaps correcting them, grouping all your reads into the families, which you can do because you have unique molecular identifiers and strand-defining elements, and then taking those groups to create consensus. And it's through the act of consensus making that we're able to identify and mark those that we think are artifacts and errors so we don't report them downstream. So in the middle of this flowchart is where we get highly, highly accurate sequencing data. Then we move it on to post-processing. You might have some post-processing that you need to do. Let's say you've got contamination. You're going to want to deal with that. If you've got some data prep that needs to occur before you can feed it into a variant caller, you're going to want to do that. And then once you get down to variant calls, there's a litany of things that you could do, such as annotating them, understanding what they could mean, filtering them if you need to filter them. And then at the bottom are really a lot of different types of reports that you can generate. And one thing to note about them is that they can pull from data from anywhere in this workflow. And when we look at this, it's complex. It's kind of like one layer off under the hood. And each one of these steps would be like a tool in a larger pipeline. But of course, software is like many, many layers of abstraction. So we could peel it back many, many more times, getting all the way down to the basic electronic principles of a computer. I'm not going to do that today. I'm going to keep it kind of simple. So one thing that is like curious to think about is NGS data big data? And if it is, will big data tools apply to NGS data? So let's talk about this. Illumina machines, they can produce gigabytes of files per day per run. So 2,000 gigs is extremely common. We have to deal with that all the time. And when you're working at some of these large-scale sequencing centers, it can be very overwhelming. Advanced algorithms are also needed to interpret this data. It's fuzzy, it's noisy, it's messy, there's technical errors. We need very complicated algorithms, and they have to run quickly. That's one of the hardest parts, actually, because you can brute force almost anything, but are you willing to wait 10,000 years? No, so we have to be clever. But one thing that is different about NGS data compared to what you hear about as big data is that in order to interpret it, we're often ignoring most of it. We only care about very, very specific things about it. So we'll take large amounts of sequencing reads, large amounts of cells, genomes, and then we'll reduce them down to just a handful of variant calls. So I think in a couple of the slides previously, some of those samples only had 100 or 1,000 variant calls. So it's decently easy for a human to understand that. So sure, we start with big data, but we end with small data. And I think that makes it a little bit more tractable and accessible for everybody. So how do we analyze and visualize these reads and consensus sequences of reads? And I'm going to show you schematics of how I look at these things. And At the end of the story, we'll track that mutation through from beginning to end and show you how it ends up in all those plots that everybody has been showing you. If you're familiar with IGV, it's a program you can download to load in read data. It's pretty easy to do, and there's a lot of great resources for how to do it. But what I'm showing here right now is one read pair in gray. Gray means that every base in this read that is gray is consensus with the reference genome, and that reference genome is right down here in kind of candy cane color. And we like to use colors because if it was letters, you wouldn't be able to read them, but the colors correspond to each of the 4 bases or nucleotides. And then in a read pair that's mapped or aligned to the reference genome, you can have a lot of different features and characteristics that describe it and how that alignment has been created. So over here on the left, we have a deletion event. So in our sample, or in our patient, or in our mouse, there are missing nucleotides there compared to the reference genome. Similarly over here, we can have an insertion. Because we're viewing things linearly in reference genome coordinates, we can't really see the insertion because it's not in that coordinate system, so we just mark it and say 4 bases extra there. But then you also get things that are really easy to understand, which are base substitutions and perhaps multi-nucleotide substitutions. And that's not all. It does get a little bit even more complex. You can have these read pairs sort of like, I guess this is topology, but they can be shaped in different ways. So we've got a perfect pair here up on the top. They're facing each other and they're clean. There's no artifacts, there's no true mutations, there's no clipping. And that looks good and simple to understand and reason about. We can also have our read pair from earlier that does have some mismatches. And we don't know if they're mutations yet. I mean, they could be artifacts. We haven't really determined one way or the other. It's just data at this point. And then down here, we have processes to soft clip the read ends. Clipping can sometimes be done to data in which parts of the data is really, really usable, but parts are not. You can have algorithms that go and clip out the part that's not, so you're not throwing away the whole thing. And then to make matters even more complicated is, as you're sequencing a molecule, first from the left-hand side of it and then from the right, if your sequencing direction is longer than the overall length of the molecule, then you'll end up with overlapping red bases in the middle. Now, you don't want to double count those because it's not fair, so you have to handle that as well. Visualizing the duplex consensus making process is kind of shown in these 3 panels. And I'll highlight first that to create a group of molecules that can then go on to be made into a duplex consensus, we need to use all those elements that Jesse had talked about in a computer algorithm so that we can apply the consensus making algorithm over each unit one at a time. The first thing that we need to do is take all of our raw sequencing reads, and they're all in different colors, so we don't know where they came from. And if you really were to zoom in, there's a bunch of mistakes in there. We take those and we group them by inferred shear points, which are basically where they align in the genome. Because if you've got a bunch of reads from chromosome 1 and a bunch of reads from chromosome 2 and they share the same tags, we know they're different because they came from 2 different places in the genome. So it's not a collision, so we definitely like to use the shear points if we can. We also use the tags and the strand orientations of those tags. All of that information gets used to finally take all of those reads and put them into colored bins so that we mark them for future algorithms. And it's this type of information, once it's all neat and tidy, that goes into the consensus-making algorithm. So now that we've made consensus, which are actually down here in the bottom, we can do some post-processing. And this is really a case study, and one of the most common things I end up hitting when I'm post-processing duplex sequencing data is contamination from other species or other individuals outside the cohort. And this is one such example. So this read pair right here, if you notice, it has a lot of what look like mutations or mismatches to the reference genome, when in fact, it's actually just a piece of DNA that floated into the tube or the experiment from another species in the lab. And those differences you see, they're not mutations in the individual, they're polymorphisms from another organism. Now they're all in phase, which is kind of giving me the clue that they all came from the same genome copy. So this is really dangerous because if you look, that's a lot of mutations. And if you're only finding 16 mutations to get your control baseline or some very, very low number, I've already found 16 right there. So we have to get rid of them. One good way to do that that we found is using fingerprinting of the species. So we can slice each of the reference genomes for all the species into tiny, tiny little bits called k-mers, and then we can use these k-mers to match, it's almost like a fingerprint for the species. We do the same thing for all of our reads. We turn those into k-mers, and then we compare the sequences to each other. And the algorithm does get pretty advanced here, but it works great because it's very, very fast and accurate. But why is this such a big deal? Well, common sources of contamination are all listed here. We've seen so many weird things, like with duplex sequencing and that accuracy, we've got a responsibility to be extra clean, even more than you probably would have done in the past. So we found things like cow DNA from BSA and culture media. And cow, they share a lot of genes with us that are highly homologous, so that's a concern. Model organisms from shared equipment is another very common way in which you can see contamination. And then previous samples after insufficient cleaning of surfaces and tools, and that could be another species, or it could be another of your same species. So how do we relate variant calls to sequences to mutations? So I'm going to give you some diagrams about once we identify a difference between a read or a duplex consensus to a reference genome, how do we know it's real? How do we interpret it? Should we call it a mutation? Should we call it something else? So I'm going to give you a few examples here. These gray bars in the middle, those are the reads from before. And then the little circle dots are a mismatch to the reference. So it could be a mutation, but let's try to interpret some of them. In the top left here, we have a germline homozygous allele, and we can kind of infer that in this simple example because it's in 100% of the reads. So that means both copies of the genome and all cells have that mutation. So we expect to see it at 100%. A germline heterozygote allele at 50% will look like this. So you'll see it in about half of the reads because they're coming from only one of the genomes in all cells. And this is all for diploid organisms. In the top right, you might have those subclones and clones. They can come in multiple copy numbers. So I've got one here that's 3, and I've got one here that's actually quite large. Now these could be expanded driver gene mutations, or they could be passengers that have expanded along with the driver genes. Or an alternate hypothesis is it's just random chance that you've gotten the same mutation more than once in your samples. They could co-arise, and interpreting these can be tricky. I do kind of highlight just this little somatic singlet because if you see something once, it's an interesting thought experiment with the accuracy of duplex sequencing. You have to wonder, if I keep sequencing more and more molecules from the sample, will I ever see this mutation again? Well, it's plausible that it was only ever found in one cell, and you just went and consumed it with your reaction, and you won't find any more copies of it. So that can sometimes happen too. How these relate to the figure that Jesse showed earlier in the day, and drawing a line from the biology on the left to how they look in the data, is just like this. We've got our large clone here. That would directly relate to this clone here. We've got a small subclone here in orange, and that kind of looks like that guy right there. And then when you get these little one-offs, perhaps, they might look like this. Okay, so we've got our mutations. We kind of know how to interpret them and think about them, but now we want to generate some basic statistics like mutation frequency. And I'm doing two very simple models for this, kind of a minimum independent events assumption and a maximum one. So for the minimum independent events assumption, we assume that every variant at a site in the genome arose from a minimum of one event and then clonally expanded. So anytime we see another copy of it, we just assume it's part of that same original progenitor clone. For the maximum, we would assume that they're all unique. So we would assume that they all co-arose, which would be the other side of that same sort of assumption. So an example of these two playing out is if you've got 100 bases that you've sequenced at one site in the genome, and most of them are adenines, but you did find 5 of them that are thymines, you could either have a mutation frequency of 1% or 5% with this model. And depending on which one you might choose, would be based on your experimental design. Now the truth probably lives somewhere in between the two, but this would be experiment dependent. The one thing I will mention here is that if you go with something that leans more to the left, is that you are going to be more protected from jackpot biases. Rapid clonal expansions that really were clonal expansions won't necessarily affect your math all that much. However, if you continue to use something that's on the left and you're working in saturation mutagenesis experiments, you might underestimate your mutation frequency. So it does take a little bit of thinking about which one of these to use, or if you want to develop a more sophisticated model in the middle. But that's for a single site in the genome, so I'm going to walk you through a couple slides of how to do this for a region of the genome. On the x-axis is our reference genome, and I've done 10 duplex sequencing reads on the left there. Where there's a hyphen, that is just simply a reference call. So it's a base that we called, and it's identical to the reference genome, so we don't care about it for right But then I have obviously colored bases. Now these are mismatches to reference, and I want to try to go through and interpret them. Well, first up, I know that the first thing I want to do is I'm interested in somatic mutagenesis, so I'm going to go and try to find all my germline variants. In this simple model, I can pick them out pretty easy. 50% and 100%, those are my hets and my homozygoses, so I can ignore those. There are other ways of finding these. You could do tumor normal calling, or if you have the genotype information ahead of time for like an isogenic line, then that can be relied upon. So we're going to ignore those for the math, but we're going to keep them into the tally of all the bases that we looked at, so they're part of the denominator. And then we're going to go and look at every time we found a mutation. Now, if we take the minimum method, where everything arose from one independent event, we really only have 5 mutations. So we've got a T that perhaps expanded once. We have an A that we didn't have any evidence that it did, same with the G and the C, and this G, though, 3 copies. With that, we end up with a mutation frequency of 1.4%, and if we were to assume these were all independently arising, that number would go up to 2.3%. So what are some things that we can do with these now that we found them? And this should look very familiar to the data that you've already seen this morning. We can measure mutation frequencies strictly and then compare them to other samples. So in this data set, which will be presented later, I think, we did a dose response curve, and we were actually able to see really tight grouping of mutation frequencies per dosage. And in this experiment, at least, we used the minimum method to protect from any clonal biases. Another thing you can do is something more qualitative. So that first slide I showed you was quite quantitative. This is unsupervised hierarchical clustering. We basically take a computer, we don't really tell it anything about the data at all as far as priors, but we load in all of our spectra and we say, could you please build a phylogenetic tree for me of how this data could look based on how it relates to itself. So it's like an all versus an all comparison type of thing. And when you look at one of these on the other side, you may be able to cut the tree in very logical places, and there's algorithms for doing this in sort of a non-subjective way, but if I cut my tree here and I cut my tree here, and I end up with 2 branches in actually both examples, in the first case, what I end up getting is a perfect split between, in this case, this is the urethane-treated group, and then on the left, it's the vehicle control group. That looks great and beautiful, and we've made a qualitative model that can actually tell the difference between a vehicle control sample and a urethane one. It gets a little more muddy down here. It's because the vehicle control, the endogenous exposure processes look kind of like BAP, and you can see that. We isolate E and U here on the left, and that looks great and clean and separated, and you can even see it in the fact that the tree is really squashed. It means that it's very different than the things on the right. But over here on the right, we have kind of a muddling of our mutagen plus the control samples. And statistically, you would say that it doesn't actually split out into its own category, but even from a qualitative point of view, there's something interesting here because a lot of the vehicle control ones are on the right, and then it trends over into BAP. So there are definitely overlapping groups that share a lot of similarities, but at either end, they're more unique towards their treatment class. And then finally is trinucleotide spectra. So you can take those base substitutions, compare them with the 5' and 3' adjacent bases. This gets you more data. It gets you more, it's a higher dimension. It actually splits your data. So if you start from a lot of data, as every time you break it down a little more, it becomes spread out. So if you want to go and do analysis like this, you'll need a lot more mutation counts in order to really resolve a true picture of what you're trying to look at. So I hope I showed you some really interesting, views on how the read data look compared to, typical views. I will say, I've been doing this work at TwinStrand now for a while, and my passions are in mutagenesis and everything somatic, but also software engineering, which I didn't really talk about all too much. So if you want to nerd out about that after the talk, I will allow it for sure. But things that I get to do now is I get to build some of these experiences for the company and make bioinformatics more accessible, try to explain this stuff, and give you all tools so that this is just easier because it doesn't need to be hard. These are my conclusions. Really, the biggest one here is that NGS data might at first appear very large and big and a bit scary, but it does get reduced to small, manageable data sets that you can interpret. and that duplex sequencing is incredibly sensitive. And I showed you a couple ways in which we have to think about it really, really carefully, where in other technologies maybe you don't care as much because the rounding error is less than the technical error. But with us, if we have like an off by one error in our code, or we make a very, very, very tiny assumption, that all could be signal that is 10,000 times higher than what biology is giving us. So we've got a higher level of attention to detail when developing with these technologies. I think that's it. Yeah, and then TwinStrand is really passionate about a science and people first approach, so we're looking for collaborations, and if you've got any cool ideas, we'd love to hear them.